Emma, did you know that researchers currently, right now, are growing human embryos in the lab?
What?
It's like something out of science fiction.
It is something out of science fiction for sure.
Nope, but it's really happening. People have been doing this for decades, actually, since like the 1970s.
So, yeah, how do they...
It was actually really complicated to figure out. So you can get the eggs and the sperm pretty easily, and then you mix them together. It's called in vitro fertilization, which you probably heard that term before. But then the tricky part was figuring out, after you've made that one cell, what are the right factors that you need to give it. So people spent years and years putting things in, taking things out, trying to figure out what's the right concoction of nutrients to give this cell, this growing cell. But after we figured that out, just add it on and let it grow.
Wow.
Like I was mentioning, this has been done in the past since the 1970s. We've known how to culture human embryos, but recently there's a news story published in NPR that a lab in New York actually has been able to make these embryos from human embryonic stem cells, which is really exciting.
Oh, wow.
Yeah.
Because with human embryonic stem cells, they're like a cell line that's been established from a previous embryo. So they're pretty far removed, but they're still the same genetic material.
So it's a little different from taking an egg and mixing a sperm together. This is, like, as Emma said, a cell line that people are using anyways. So it has a slightly different origin. And this gives scientists like a lot more flexibility.
Yeah.
People have been able to get access to human embryos from in vitro fertilized eggs pretty easily because we're already making those for the purpose of like helping people get pregnant. The first IVF baby, Louise Brown, was actually born in 1978.
So that's a pretty long technology that they've been using it.
Yeah. So we've been able to grow these things for a while. But when you do in vitro fertilization for the purpose of pregnancy, you only really culture those cells for 6 days. But in the lab, we're able to go up to 14 days. And then there's some guidelines in place where there's a hard stop at day 14. And we'll kind of get into that later and the reasons behind it.
Do couples have to consent for the researchers to use the leftover embryos? And...
Yes, yeah, absolutely. So in the actual paper where they publish these results, they don't call it a human embryo. They call it a pre-gastrulation human epiblast.
Sounds like a very PC way of saying it.
Yeah. Of course, all the news stories are saying human embryos.
Yeah.
But there's not really that much difference between embryo and epiblast. They're kind of like, there's a little bit of a technical difference, but pretty similar. And the pre-gastrulation means like before the gut develops, which happens around 3 weeks. So just trying to say that this is a very early embryo. It's not like they're letting it grow for weeks and weeks.
Right.
For most of the experiments that they did, they only really let it grow for around 4 days. And 4 days in their system was similar to what a human embryo looks like at 10 days.
Oh, wow. So it's almost like an accelerated embryo program.
And their system is pretty cool. So in the past, and we mentioned this in a previous video, people like to grow cells in 2D, meaning that they're just flat on a surface, side by side. But for the system, they took single cells and they actually embedded them into this gel so that they can form that 3D structure that we talked about that's so important for cell identity and morphology or shape. Using this system, they were able to make something that looks pretty similar to a real human embryo. And why are they doing this, of course?
Right.
So this lab is really interested in symmetry. Symmetry is important for an embryo because you need to break that symmetry in order to start organizing yourself into compartments and making all the tissues that you need. And the reason for this is that when you think about it, every cell in your body has the genetic information to make all the things. Your skin cells have the same information that your brain cells have. So what makes it different? That would be gene expression. You have the information, but you're not actively making it in every cell type. So in order to have these differences, there are these developmental cues called morphogens that help your cells figure out which genes to turn on and off. And if your embryo has symmetry, if those morphogens or those cues are just present everywhere, how do you know how to make a brain cell here and a skin cell there?
And a lot of times those morphogens will come out like if you define where the head is versus kind of the posterior part of the body, they'll kind of go on a different gradient. So to produce, like the spinal cord, I think works that way, and there's other morphogens too, but they have to be expressed at a very precise time in order for you to develop properly.
Exactly. So this lab is really interested in like kind of that first moment where you break symmetry and you start to make that gradient that Emma mentioned. One of the questions they have about this is, can the embryo establish that gradient by itself, or does it require cues from mom, right? So they were able to show that just by adding one morphogen called BMP4, that they could establish the gradient without mom. Sorry, mom. We don't need you. So something that they were really excited to see in these embryos is that they developed what's called the primitive streak. And the primitive streak is when the embryo starts to elongate and break that symmetry. So this is like a total landmark for developmental biology. But it also brings us to an ethical question.
Lots of ethical questions.
Yes. So currently the regulations on human embryo research, currently there are no official regulations on embryo research in the US. In 12 other countries, including the UK, there are like strict laws.
I didn't realize we didn't have strict laws.
Yeah,
I'm pretty surprised.
It's more of a guideline, actually.
Wow. I mean, considering how CRISPR babies blew up.
Yeah. It's a guideline that, I mean, everyone respects, but it was something about, like, in the 1970s, you know, when we first started doing IVF and everything, they made, like, a panel of researchers and doctors to try and determine, okay, slow down, like, we need to think about ethics here. And that panel decided that we should stop at 14 days. And the reasoning for this were 3 main points. First of all, by 14 days, half of the embryos won't survive anyways. Like, miscarriage is actually very common. So they argued that we have less of a moral obligation if 50% of these weren't going to go on to make a baby anyways. Second is that the embryos can actually become twins before 14 days. So they kind of argue that the embryo doesn't have a biological identity at this point, since it can become one person or 2 people. And the third, probably the most important, is that by 14 days you develop that primitive streak that we talked about. So the embryo is really starting to organize into something. At the time, Jimmy Carter was president, and there was some staff turnover, so the report just got like forgotten about. And nobody ever made official regulations based off of it. Yeah. So there's no laws, but we stop at 14 days. And there are like 4 other countries like us that follow the same guidelines but don't have laws. And there's 12 countries that have strict laws following the 14-day rule.
But there is still a lot of controversy behind human embryo research, especially since we have the induced pluripotent stem cells. But in a lot of cases, those don't act the same as the human embryonic stem cells. But there still is. I think people can be pretty divided on the topic, and a lot of it just depends where you believe life begins, and that kind of drives if you're willing to use embryos or if you're not willing, and that's kind of the hard thing when it comes to making laws, because a lot of it's driven by emotion and religion and all of that.
And your personal beliefs. And then the other thing, you brought up induced pluripotent stem cells. So now that they've made these human embryos with human embryonic lines, I'm sure someone's going to try to make them with induced pluripotent stem cell lines. I mean, that could open a whole new door using patient-specific. What if you're having fertility problems and they can make induced pluripotent stem cells from you and model the embryo from your cells?
And then you wonder about epigenetics with that, because in the embryo there's so many epigenetic changes that are happening based on your environment. So would that be able to happen with the induced pluripotent stem cells? Yeah.
I mean, there's so many questions that my scientific brain wants to answer, but it's still, even if it's coming from an induced pluripotent stem cell versus a human embryonic line versus an actual in vitro fertilized egg, you're still making something that's pretty close to a human embryo. So at what point is that real, and at what point is it ethical to keep growing it, or at what point do we have to stop it?
Yeah.
The group at New York, at Rockefeller University, that developed this model actually argues that you shouldn't consider this a real embryo because none of these would be able to go on. None of these would probably practically survive if you were to implant it.
It's probably true. But
Yeah.
That could be
Probably true, but I don't know.
kind of a slippery slope to go down.
Yeah. Because of this recent publication, the International Society for Stem Cell Research is getting together to reassess that 14-day rule and update the guidelines now that
they haven't been updated in like 35 years.
Yeah.
It's pretty insane.
Yeah.
So.
But now, do we need to also consider, do the guidelines state that this is specifically for in vitro fertilized eggs, or should we add embryonic stem cell lines and also make that hard cutoff at 14 days? Do we still want to have the cutoff at 14 days? Should we go longer? There's still like a total black box of what happens after 14 days.
Yeah.
So.
Well, a lot of this work is being defunded too at the moment.
Yeah. So I don't know about the embryonic.
I think that's still okay. It's more like a fetal tissue.
So a lot of groups will use fetal tissue that will be discarded for their research to learn about like gene expression at certain stages, if you get fetus at 3 weeks versus 4 weeks versus 5 weeks. Recently, there are regulations passed that, you can't receive federal funding for this kind of research.
So, yeah.
Anything involving fetal tissue. So this is pretty controversial. I mean, a lot of people argue, like for moral and religious reasons, like we shouldn't be doing that kind of research on humans. Other people argue, well, this is really sad, but this fetus is going to be discarded anyways. Wouldn't it be great if we can learn something from this life and make it mean something if it's going to be discarded?
And then other people say like, oh, well, isn't that like an analogy of, someone told me this once, of like if someone robbed a bank and then they had lots of money fall, if you picking up that money, would that mean you were also like aiding and abetting and robbing the bank? So there's a lot around this.
Yeah.
And just, it's interesting to follow the discussions, but a lot of them are not very productive.
That's so true.